Hello students, today we are going to discuss chapter 9. It's a short revision, biotechnology principles and processes. In this session we are covering all this chapter, so you don't need to spend more time in this chapter. I am here discussing most of the important points here. So let's begin. This chapter is divided into 3 parts: the principle of biotechnology, tools of recombinant DNA technology, and processes of recombinant DNA technology. We know the basics of biotechnology, why we are using biotechnology. This is for producing the genetically modified organism, and in vitro fertilization leading to test tube babies is also using this kind of methods, so on. So we are not going to emphasize these kind of points, and there is given a definition for biotechnology: the integration of natural science and organisms, cell, parts, therefore thereof, and molecular analogs for products and services. That's the definition of biotechnology. Let's go to the principles of biotechnology. There are mainly two principles, two core techniques: genetic engineering and bioprocess engineering. There are two core techniques: genetic engineering and bioprocess engineering. In genetic engineering, the DNA or RNA, the genetic material, alters and it is then introduced into a host organism. So the phenotype of the host organism changes. That's in genetic engineering. In case of bioprocess engineering, it's used for producing antibiotics, vaccines, and enzymes. So here include the maintenance of sterile microbes or eukaryotic cell in large quantities for the production of antibiotic, vaccines, enzyme, etc. By genetic engineering, we can produce the organism with a desirable gene. So next paragraph they are discussing about sexual and asexual reproduction. In case of asexual reproduction, we can preserve the genetic information, while in sexual reproduction there is chance for variation. So by this traditional hybridization methods or reproduction method, there is more chance to get the undesirable gene. So we are using biotechnology, that is recombination DNA method, gene cloning, and gene transfer. The three methods, what are they? Recombinant DNA method, gene cloning, gene transfer. These three methods are mainly used for producing the desired genes. Next we are discussing about the rDNA technology, which is based on cloning. The steps for producing artificial recombinant DNA molecule. There is given 3 steps: identification of DNA with the desirable genes. First we are identifying what kind of DNA we need to recombinate. Introduction of the identified DNA into the host. First we are taking the desired DNA, then we are introducing this DNA into plasmid. We know that plasmid is a autonomously replicating circular extrachromosomal DNA. Plasmid can replicate autonomously and it is a circular and extrachromosomal DNA. Okay, first this process is done in Salmonella typhimurium species by Stanley Cohen and Herbert Boyer. In 1972. So they isolate antibiotic resistant genes. Suppose this is the gene and this incorporated into the plasmid. So this is incorporated into plasmid. The cutting is done by restriction enzyme, and this after cutting the desired DNA, it's then cloned. It's then attached to the plasmid, and this plasmid act as a vector. And This restriction enzyme is known as molecular scissors, and this DNA is joined using DNA ligases. So we get the combination of circular autonomously replicating DNA. It is known as the recombinant DNA. So we can transfer this vector into the host plant. So from this host, this vector is multiplying, and it must have a replication or ORI, origin of replication. It helps for starting the replication in an organism. And the ability to multiply copies of antibiotic resistant gene in E. coli is known as cloning. Okay, and the last step is maintenance of introduced DNA in the host and transfer of the DNA to its progeny. These are the basic steps. Then next we can go to the tools of recombinant DNA technology. First one is the restriction enzyme, and there are two enzymes. First one is the E. coli are isolated from E. coli are one that add methyl group to DNA, and the other cut to DNA, and this is known as the restriction endonuclease later. And the first to identify the restriction endonuclease is Hint2. Hint2 cuts the DNA at a particular sequence, and that specific sequence is known as recognition sequences. Hint2 cut DNA at a specific sequence, and that specific sequence is known as recognition sequence, and it is a 6 base pair recognition sequence. It is a 6 base pair recognition sequence that can identify by Hint2. And there are some other restriction endonucleases. We know around 900 restriction enzymes that are isolated from over 230 strains of bacteria. And let's discuss how to name these restriction enzymes. The another example for restriction enzyme is EcoR1. EcoR1, the E stands for the genus name Escherichia, and CO stands for the species name coli, and the letter R is the strain name, and the Roman letter 1 represent the order in which the enzymes were isolated. Okay, first strain is genus name, second two letter is species name, and the third letter is the strain name, and the Roman number is the order in which the enzymes were isolated from that strain of bacteria. Okay, these restriction enzymes belongs to a larger class of enzymes known as nucleases. There are two kinds, exonucleases and endonucleases. Exonucleases cut the DNA at the end, and endonucleases cut the DNA to specific position, that is inside, not the exit region, at a specific positions. So this endonuclease can function as functions by inspecting the length of the DNA sequence. They can inspect the length of the DNA sequences. Okay, and these restriction endonucleases cut the double-stranded DNA into two pieces. They cut at the sugar phosphate backbones, and these restriction endonucleases can recognize the palindromic endonucleotide sequences. Palindromic nucleotide sequence. What is this palindromic nucleotide sequence? Palindromic nucleotide sequences give the same word when it read forward and backward. For example, example is this Malayalam. It can read the same from forward and backward. Like that, the following sequences reads the same on the two strands in 5' to 3' direction. That is, this is same. GAATTC is same as when reading 5 to 3 GAATTC. So this is a kind of palindromic sequence. So here we are, this diagram is the action of restriction enzyme. The restriction enzyme cut the vector DNA and the foreign DNA. Here it's the action of the EcoR1. EcoR1 cuts at G and A between the guanine and adenine bases. So remember these bases, it makes cut, so it forms a sticky end. So based on this sticky end, ligases can function. So these are joined by ligases and forms a recombinant DNA. Okay, that's all in this session. And this sticky end is named because they form hydrogen bonds with the complementary cut counterparts. Okay, then this is the same diagram that we discussed. This is a foreign DNA and vector DNA or plasmid, and make a cut here using the restriction enzymes, and the same restriction enzyme is used for cutting the foreign DNA and the plasmid DNA, and the foreign DNA is also cutting using restriction enzyme, and this is joined to the plasmid. Plasmid is a extra chromosomal DNA. It's autonomously replicating and it is circular, and ligases join and forming a recombinant DNA molecule, and this DNA recombinant DNA molecule is transferred to bacteria or E. coli, that is the host cell, and this host cell is divide by using ORI, origin of replication. So the replication start from the ORI. Okay. By the use of restriction enzyme, the DNA become the fragments, and these fragments can be separated by using gel electrophoresis. The methods for separation of the DNA fragments used is the gel electrophoresis, and this work based on the size of the fragments. The smaller fragments can move long distances, and it's run in agarose gel. Agarose, which is extracted from it's extracted from seaweeds, and the DNA is negatively charged, so it's moved towards the positively charged electric field. Under electric field, it moves towards the anode, which is positively charged, and this can be visualized by using staining. The stain which is used is ethidium bromide. It's important. Ethidium bromide is the stain, and agarose is the gel, by exposure of UV radiation. And you can see the bright orange colored bands of DNA in ethidium bromide. It's seen as a bright orange color. Okay. This separated piece of DNA can be extracted from the gel, and this process is known as elution. Note that elution, the extraction of the fragments of DNA from this agarose gel, is known as elution. And this purified DNA fragments can be used for the constructing recombinant DNA, so that can reuse. Next is the cloning. The mainly using cloning vectors are the plasmids or the bacteriophages. And what are the features of the cloning vectors? They must have the origin of replication, selectable marker, cloning sites. Okay. First one is origin of replication. So from the origin of replication, replication starts. It's very important one. So it's also responsible for controlling the copy number. Second one is selectable marker. Based on the selectable marker, we can identify the recombinant DNA. So for example, this selectable marker is present only in the recombinant DNA. So the main using is the genes encoding resistant to antibiotics such as ampicillin, chloramphenicol, tetracycline, kanamycin. These are the main antibiotics used: ampicillin, chloramphenicol, tetracycline, kanamycin. Suppose there is an E. coli which is resistant to antibiotic, and that is made by rDNA technology, and some E. coli is not, this is the E. coli formed by rDNA, and this is not the recombinant DNA. So and it has a ampicillin resistant gene. So it doesn't have ampicillin resistant gene.